If you think of anything later, feel free to ask, and keep in mind, you can always ask as we go through the lecture material. I know some people don't like to, some teachers don't like you to ask questions, but if you don't understand what we're talking about, always interrupt so that you can get point A before we move on to point B. If there aren't any questions right now, we're going to go back to plant tissues. And plant tissues, and in particular how vascular tissue works, is going to be our last topic. So we'll probably finish up with plants either today or Monday. So Monday we'll have some questions on plants posted, and they'll be due not next Monday, but a week from then, whatever that date is. Today's the 9th, so that would be maybe the 19th if I'm counting right. So keep an eye out for those. We've been looking at the vascular tissue and how it's arranged in stems and roots. And when we left off, we were talking about herbaceous dicot roots. And herbaceous dicot roots are roots and stems that are found in non-woody plants. So I'm sorry, I said root. This is the stem in a sunflower. So let's magnify this and let's focus in on one of those vascular bundles. And we would see something like this. So here's our vascular bundle. This kind of, I don't know, reminds me of an acorn. So this kind of acorn-shaped structure. In here, in the lower part of our photograph, these are parenchyma cells. And since they're inside the xylem and phloem, these parenchyma cells make up that area that we call the pith. Out here at the very edge of our stem, we have a single layer of very thin cells. These are epidermis cells. Now, out around these epidermis cells, we have some ground tissue. Here, the ground tissue is mostly parenchyma cells. But if we would look at some herbaceous dicots, we do find some collenchyma here too. So we want to keep that in mind. And then we have ground tissue kind of in here between the acorns, between the vascular bundles, connecting the cortex out underneath the epidermis to the pith in the middle. So if you would look overall, there's more ground tissue, there's more parenchyma in this plant than anything else, but we're going to focus on a vascular bundle for right now. Well, one of the keys to keep in mind is when we stain plant cells, the stains that we use generally attach to the chemicals in the cell wall more than other parts of the plant cell. So cells that look very dark tend to have a relatively thick cell wall compared to the cells around them. So we see some dark cells. I'm focusing on this acorn. We see some dark cells in here, kind of the bottom part of the acorn. This band of dark cells, or here's one over on the left that's a little bit larger, these are our xylem cells. Now just to the outside, so that would be moving upward on our diagram, just to the outside, there's this kind of light greenish above the purple. This light greenish is the phloem. Remember that when we look at a cross section of a stem or a root, the xylem is always inside the phloem. The xylem is more towards the middle. And then if we go just a little bit beyond this light greenish, we see a kind of a darker green out here. And this darker green is sclerenchyma cells. So these are the three main types of cells in here. There is a single layer of vascular cambium right between this purple and this green that makes up the phloem. We'd have to magnify our picture a little bit better, or use a different stain to see it very clearly. So when we think about a herbaceous dicot root, the vascular bundles are arranged in a single circle, and that circle is usually pretty close to the periphery, to the edge of the plant stem. Now, the other type of stem, which we've looked at once or twice, is a woody dicot stem. And if we look at a woody dicot stem, the key is if we look at the xylem makes complete circles around the stem. The xylem is the material that we call the wood in a woody plant. And we can divide this wood up into two basic categories. Remember that the vascular cambium is on the outside, right between the xylem and phloem. And that means the newest xylem is close to the vascular cambium. So the newest xylem is towards the bark here. The oldest xylem is in towards the middle, towards the pit. The newest xylem, typically maybe 5 or 6 years, will have cells that are actively transporting materials throughout the plant's body. So this last 5 or 6 annual rings, if we're looking at a tree from around this part of the country, is sometimes called sapwood. Sap is kind of a very generic term that we use for the fluid flowing through the vascular tissue. However, if we're looking at a tree that's, say, 100 years old, the sapwood is only about the last 6 years or so of xylem. The first 94 years are heartwood. As the xylem is transporting material around the plant's body, you know, think of it like a person with a bad diet, your arteries clog up, the xylem clogs up. And so the inner layers of wood, the heartwood, don't transport water or materials dissolved from the roots anymore. They're clogged up. They're, at least in terms of transporting materials, they're non-functional. Keep in mind the wood, and since wood is xylem, the xylem plays a very big role in holding the tree body up. So we don't want to think that it's not important just because it's no longer transporting water. That's not the case. When we think about monocots and dicots, we've been talking about them quite a bit lately. There's a lot of ways to tell a monocot and a dicot apart. Now notice when we say to tell a monocot apart from a dicot, we're really talking about a plant called a eudicot, EU dicot. Now remember, U is the same prefix that we find in eukaryotic. U means true or real or actual. So eukaryotic cells have a real nucleus. The karyot is the nucleus. So eudicots are the true dicots. And the reason that we call them eudicots a lot of times nowadays is when people look at the flowering plants, they divide them up into about 10 different categories. One of those categories is monocots. All the other categories are dicots. And they used to just all be in one group, the dicots, but now one group has been separated out and called the eudicots. And then some of the other things that used to be dicots have been put in very closely related categories. So the monocots branch off one direction, the dicots and all the other little groups related to them branch off the other. Notice that the monocots were the last group to evolve. So really, if you go back to the times of the dinosaurs, we don't see broad plains like the Great Plains of the United States or the savannas that we find in East Africa. Those type of ecosystems didn't exist because there weren't monocots yet. So we didn't have grasslands since most grasses are monocots. So let's think about some ways to tell them apart. Well, one way to tell monocots apart from dicots is monocots tend to have very thin, elongated leaves. So think of a blade of grass, or if you know what the leaves on most bulb plants look like, things like tulips and lilies and daffodils. Dicots are usually what we call broadleaf plants. Compared to the length of the plant, of the leaf is relatively broad. Now, monocots and dicots actually get their name from the seed parts. Monocots have one cotyledon. Here, what's shown is basically a diagram of a corn kernel. Corn is a monocot. A cotyledon literally means a seed leaf. And when a monocot first germinates and starts to grow, it produces one seed leaf. And the seed leaf starts photosynthesis and helps the baby corn plant survive until it can make more leaves of its own. Now notice that it's not always easy to tell whether a seed is a monocot or a dicot. You might think about a corn kernel and you might say that's pretty obvious, but some dicots, the parts are so tightly clasped together that it's very difficult to separate them. A good example of a dicot that it's easy to see it's a dicot is a peanut. If you open a peanut shell and you separate those two halves of a peanut, those are the two cotyledons. So when dicots germinate and start growing, they make two seed leaves. So because of that, they're very easy to distinguish. If we look at the leaves on a monocot, the veins are parallel. Remember, I said the veins are all those ridges you can see on a blade of grass. If we look at the veins on a dicot, they're branching. If you think of that main vein that actually runs through the petiole and connects the leaf to the plant, it looks like a tributary system in a river. Now keep in mind, some dicot leaves are very small, they're microscopic. So when we say broadleaf, we're really comparing the width to the length. We're not talking about the overall dimensions. If we're looking at a leaf that's only 1/8 of an inch long, it might be a 1/16 of an inch wide.
This would be the easiest way, what I
But
said.
it's still considered to be a broadleaf plant.
Right?
Now,
Well,
if we
hopefully there won't be,
Yeah.
think
What
about,
do you think the percentage is?
but he's been at,
Oh, sorry.
if we think about a stem, and notice we could do something similar with roots. Remember, we talked about roots the other day. There's a difference in how the vascular bundles are arranged. So in a monocot, so this would be something like that corn, the Zea mays that we looked at, the vascular bundles are scattered all the way from towards the middle of the stem out towards the periphery. If we look at a dicot stem, we see a ring of vascular tissue. In a herbaceous dicot, there are some little gaps between the xylem and the phloem. So it's not a continuous ring all the way around. But if we look at a woody dicot plant, then those form continuous rings all the way around. If we look at roots, monocot roots tend to have what we call fibrous roots. Fibrous roots are small roots. They have lots of branches. This is what you see in grass that might grow in your front yard. If you've ever pulled up any grass and you look at the roots underneath, they form a kind of a solid mat. There's as much root as there is soil. And this is a fibrous root. If we look at dicots, dicots tend to have taproots. There's one large main root underground, and it's usually vertical. And then there are some smaller branches off that main root. A carrot is a good example of this. Or if you've ever had to cut down a tree and then you've started to dig up the stump and you've looked at the root, you've seen part of the taproot of that tree. And finally, we can also look at flower parts. So when we say flower parts, keep in mind we're talking about those sepals, petals, stamens, the stigma at the top. Monocots tend to have flower parts in multiples of 3. And usually 3 are 6. Anything more than 6 is very rare. If we look at dicots, dicots tend to have flower parts in multiples of 4 or 5. And again, sometimes 8 or 10, but anything bigger than that is very unusual. There are no plants that I'm aware of that have flower parts as a multiple of 12. So because notice if it had a multiple of 12, you wouldn't be able to figure out whether it was a monocot or a dicot. So usually we can count the flower parts, and assuming we have a flower that isn't damaged, we can figure out pretty easily whether we're looking at a monocot or a dicot. Now, keep in mind what we said up here. Besides the stem, we could also look at the pattern of vascular tissue in the root. And we could also look at the pattern of mesophyll cells in the leaf. So there are lots of different ways to tell monocots and dicots apart. Economically, they have very different functions. Think about the monocot here. Remember what I've said. Monocots include things like grasses. So all the plants that we refer to as grains or cereals are monocots. So corn, wheat, rice, things like that. So as we go around the world, these plants are incredibly important. If we look at the world diet as a whole, most of the calories that people eat come from monocots, come from cereals or grains. If we look at dicots, this is pretty much everything else in your diet. Not meat, of course, but all the other plant material. So when we're looking at things like tomatoes, watermelons, potatoes, peanuts, beans, things like that, these all come from dicots. And notice we do eat lots of different parts of the plant. If we look at flour that we get from wheat, or in some parts of the world, they make it from rice or other materials. Flour is basically the ground up seed. So we're taking the seed, we're grinding it up. In dicots, we don't usually eat the seeds just by themselves. We might eat them as part of the entire plant. We might eat a seed when we eat a tomato or an apple, but we don't eat the seed even ground up typically. But notice with dicots, we also eat other parts of the plant. Some people eat the stems of plants. That's what you're eating when you eat something like celery. Some people eat part of the flowers. That's what Brussels sprouts are. If you eat cabbage or lettuce, you're eating part of the leaves. So we can eat different parts, but for monocots, we're mostly eating actually the fruit. That's what contains the seed. With dicots, we're eating a fruit if we eat something like a tomato, but we could be eating part of the leaves, you know, if you're eating lettuce, part of the stem if you're eating celery, part of the root if you're eating a carrot. And there are other parts that don't fit into those categories. Potatoes are something called tubers. Potatoes actually grow from part of the stem above ground, but the stem part bends down and the potatoes actually grow underground. So lots of different options, but when we eat plant material, it's usually from a monocot or a dicot. We don't typically eat plant material from conifers or from the seedless vascular plants. So from things like ferns or from non-vascular plants, the mosses and things like that. So mostly the plant food that we get comes almost entirely from monocots and dicots. There are a few minor exceptions, but not many. Any questions about monocots and dicots before we leave them? Okay, with most of the types of tissue that we've been discussing, we actually looked at the types of cells in the tissue and some things like that, but I didn't really talk about that when we just discussed xylem and phloem. So now we want to look at the actual cells in xylem and phloem. So hopefully you see some xylem cells right now. If you don't, let me know. So xylem and phloem make up the vascular tissue And when we look at xylem, there are two types of cells in xylem. Some cells called tracheids and some other cells called vessel elements, or they're sometimes called vessel members. Now, both of these cells have pits in them, and pits are just little holes. Let's look at tracheids first. And the reason we want to think about tracheids first is here are some on the far right, or there are some in the center here on the left. Tracheids are found in all vascular plants. So whether we're talking about horsetails or lycopods or ferns or conifers or monocots or dicots, all of them have tracheids. Now, if we look at the tracheids, what we see is they're very long, skinny cells, and at the bottom, they come to a point, and also at the top, they come to a point. And they kind of fit together right where the cell slants. Now, what we see is, remember, these are plants, so plant cells have cell walls. So these double lines here represent the cell walls. There are pits, there are little holes in the cell wall of the upper cell, and there are pits, there are little holes in the cell wall of the lower cell. And these pits line up. So because these pits line up, this allows material to pass from the lower cell through the pit into the upper cell. So this is how they're going to transport material upward from the roots. Now, what type of material do they transport? Water and minerals that are dissolved in that water. So mostly what's absorbed by the roots. We'll see one exception to this later. Now, you might think, okay, you say they're transporting water. What about all the cytoplasm in these cells? What about the nucleus? What about endoplasmic reticulum? Well, that doesn't get in the way because tracheid cells are dead when they're mature. All that's left of the cell is the cell wall. There's no cytoplasm. There's none of the organelles that normally we would find in the cytoplasm. There's no chloroplast, no mitochondria, no endoplasmic reticulum. There's no nucleus. So these are essentially like hollow pipes. So nothing interferes with the movement of water and the dissolved materials upward. And in a few minutes, we'll talk about how xylem works, how it actually transports material upward. And this continuous column of water between down in the roots and up in the tip of a leaf that might be 100 feet above the roots, maybe more, this is crucial. Now, the problem is these pits are relatively small. So that means there's, in a sense, a little bit of a traffic jam. Water that's moving up a tracheid, there's not necessarily a pit open for it to move through when it gets there. So vessel elements are much more efficient. Vessel elements are only found in angiosperms, flowering plants. So the monocots, the eudicots, and some of those other minor groups that are classified with the dicots. And the cells are actually pretty similar, except there's one difference, well, a couple of differences that we're going to emphasize, I guess. The tracheid cells are very narrow. If you look at a vessel element over here compared to a tracheid cell, vessel elements are much bigger. They have a much larger interior. This interior is just called the lumen. Anytime we have a structure like a pipe in a body, the inside of that pipe is the lumen. So the lumen is much bigger, which helps water to move more rapidly through the vessel elements. But there's another factor that's even more important. In the tracheids, we have these little pits that connect one cell to another. And you could think of these little pits as like sticking a needle through a piece of material. So they're relatively small. If we look at the cell wall down at the bottom and up at the top of these vessel elements, the cell wall has big, large, they're called perforations, but these perforations really, sometimes there's pretty much no cell wall left. There's just a big, humongous hole between one cell and the cell above it, or the cell below it, whichever way we're thinking of. So vessel elements tend to be more efficient. They have a larger diameter, and what we call the end walls, this part of the cell wall between two cells, has larger, much larger holes in it. So water flows much more nicely. Now, xylem pretty much only transports water and solutes that are dissolved in water upwards from the roots. There's one exception to this that we'll see later. But so let's think about how we can get the water from the roots up to leaves that might be 100, 200 feet above where we started out. And one of the key factors in moving material up the stem of a tree is something called transpiration. Now, there are several processes involved. Transpiration is what causes water in a leaf to evaporate. So we're going to be thinking of water moving up the plant, and even though the water technically starts in the roots and moves upward, we're going to start in the leaves up at the top and think about working our way down, just because you might think that the water moves upward from the roots, but it's really pulled upward by the leaves. So the process that occurs in the leaves that pulls the water up is something called transpiration. And transpiration is really just the technical term for evaporation. You know, you might sweat on a hot day. Plants transpire on a hard day. And transpiration just refers to that loss of water out the stoma that we've talked about on several occasions. Now, the second factor that's important is if we look at the tracheids and we look at the vessel elements in the xylem all the way through the stem, and even in the root, because those do extend down into the root. If we look at those, what we see is there are two processes called adhesion and cohesion. So those are going to be important as the water moves upward. So the water moves out of the plant by transpiration. It moves upward in the plant by adhesion and cohesion. And water is absorbed in the root by osmosis. At least there's one thing here we're vaguely familiar with. So let's keep in mind why we need water in the first place. The reason that we need water in the first place is, remember, water is one of the reactants of photosynthesis. So when we look at water, let's think about how we get a pull upwards. And the real key to thinking about the movement of water through the tree is something that we talked about back in Biology 105. When we talked about Biology 105, we talked about ionic bonds when we discussed chemistry. We talked about polar covalent bonds when we talked about chemistry. We talked about polar non-covalent bonds. And we also talked about a type of bond that's found in water and really just a few other types of materials. Does anybody remember the types of bonds that are found in water?
Hydrogen bonds.
Hydrogen bonds. So when we think about water all the way up in a leaf, going down the stem, going into the root, going to the point where the water is absorbed, there is a chain of hydrogen bonds connecting one molecule of water to the next molecule of water all the way up to the top. So we want to keep that in mind. all these water molecules are connected to one another. Now, remember what we've said, when water gets up to the leaf, plants aren't very efficient. Most of the water gets lost, which means most of the water gets lost by transpiration. We said in most plants, we're talking about 98% of the water gets absorbed by the roots, goes out the leaves. Only 2% in a plant that's super efficient, 10% of the water absorbed by the roots might actually get used for photosynthesis. So when this water gets up to the leaves and it actually leaves the xylem, there's two possibilities for it. Some of this water enters a mesophyll cell. Remember, mesophyll cells are the main cells that carry out photosynthesis. So if this water enters a mesophyll cell, now it can become a reactant in photosynthesis. But that only happens to somewhere between 1 and 10% of the water. The other 90 to 99% is transpired. It leaves the leaves. But what you have to keep in mind here is we have a chain of hydrogen bonds linking all our water molecules. So imagine that you jerk on that chain and you pull a water molecule out of the leaf through the stoma. Well, when you do that, that pulls all the other water molecules in this chain up one notch. So the hydrogen bonds are what cause the cohesion and adhesion. Cohesion refers to the fact that water molecules bond to each other through these hydrogen bonds. So cohesion is the water molecules attaching to each other. Adhesion refers to the water molecules can also form hydrogen bonds with some of the molecules in the cell walls of the tracheids or the cell walls of the vessel elements. So these hydrogen bonds, since some of them attach the water to the cell walls of the xylem cells, this keeps the water from slipping back downward. And keep in mind, water molecules can potentially form 3 different hydrogen bonds, one for each hydrogen atom, one for the oxygen atom. So they could have one bond to a cell wall molecule, and they could have 2 other bonds to water molecules. So this adhesion and cohesion hold the water in place and then make sure that when we lose some water by transpiration, we jerk up this whole attached chain of water molecules. Now, what happens down at the bottom in the roots? Well, keep in mind, we're constantly pulling water upward. So if we're constantly pulling water upward, that means if we look at xylem down at the very bottom of the root, xylem at the very bottom of the root doesn't have as much water in it. In other words, think in terms of something called diffusion, the water there is at a low concentration. So since the water is at a low concentration, water moves into the roots by osmosis. And it moves all the way into the xylem. So water moves into the xylem from the ground around the plant's roots by osmosis. It moves up the plant by adhesion and cohesion, and then it moves out of the leaf or into a mesophyll cell for photosynthesis because of transpiration. So when we look at this, this whole thing happens because of hydrogen bonds. Without hydrogen bonds, this would not work. And if this didn't work, then you wouldn't be able to have plants reach the height of trees. Plants would be limited to, at the most, appears to be about 4 or 5 inches without any vascular tissue. So this is really crucial. Now, if we look at the guard cells, we've said that the guard cells regulate water loss. So here we have some guard cells. We have the central vacuole, which is the blue swimming pool in the middle. We have a stoma in between them. And what guard cells do, remember we said that the cell wall on the inside is thicker, the cell wall on the outside is thinner. Guard cells have some bands of cellulose, and the bands of cellulose run perpendicular to this long axis, and they circle the cells. And what these bands of cellulose do is they stop the cell from expanding. So it can't really get any larger. So what happens is when water is plentiful in the plant, the guard cells pump in potassium ions into their central, well, first into their cytoplasm, then into the central vacuole. If we put potassium ions into the central vacuole, that lowers the concentration of water. So now water will diffuse into the cytoplasm and then into the central vacuole. So as the central vacuole swells and expands, the cell can't really get bigger. And since the cell wall on the inside is pretty stiff, the outside bulges out a little bit. And as it bulges, it kind of pulls this inner cell wall with it because of those cellulose bands. So when it pulls this inner cell wall out, the stoma opens. Notice that the stoma is opening when there's enough water to fill the central vacuole to overflowing. So what happens if the plant is living in an area where there's a drought? Well, when plants start to become dehydrated, the first place that loses water is the central vacuole. So the central vacuole loses water, it goes into the cytoplasm. If the plant's still dehydrated, that water may come out of the cytoplasm to be used elsewhere. But when the water leaves the central vacuole, we quit having this pulling outward. And when that happens, the two cell walls of the guard cells that are on the inside fold in, and the stoma is closed off. So the plant loses a lot less water. Now keep in mind, nothing is 100% efficient. So even when the stoma closed, they still lose a little bit of water, but really 1,000 times less in most cases that have been measured. So it's much more efficient. I was worried we weren't going to get to talk about chemistry this semester. So, you know, that would be tragedy. So this is showing kind of our chain of water molecules, where some water molecules, this is a sieve element here. So you can see these big perforations in the middle where the cell wall doesn't get all the way across. Some of the water molecules form hydrogen bonds to pieces of the cell wall. Some of the water molecules form hydrogen bonds to the water molecule either above them or below them. So this is what keeps our movement going through the xylem. Now let's look at phloem. And when we look at phloem, there's one main type of cell. And this cell is called a sieve element or a sieve tube. And when we look at this, in a way, the cell structure looks kind of similar to what we talked about with a tracheid. If we look at a column of phloem cells, of sieve tube cells, if we look at a column of these, the end walls, which are the little kind of salt and pepper shaker looking thing here, the end walls, which are sometimes called sieve plates, have holes in them. So material can pass from one cell into the next cell. However, even though in that sense, it might seem like this is going to work similar to the xylem, there's a huge difference. If we go back to the xylem for a minute, We go back to the xylem, the vessel elements and the tracheids are dead. When they're doing their job, they're dead. No cytoplasm, no organelles that would normally be found in the cytoplasm, no nucleus, there's nothing there. If we go down to our phloem cells still have cytoplasm. Now, they don't have most of the organelles that are found in the cytoplasm, or if they have them, they have very small amounts. So they don't have as much mitochondria as a regular cell or as much endoplasmic reticulum. And most plants, the sieve tube doesn't have any. So this is a living cell though. There is cytoplasm in here. So this means that materials are not going to be able to move through this cytoplasm the same way that they move through those tracheids and the vessels. Because that only works when we can guarantee what one water molecule forms a hydrogen bond to. Here, instead, what happens is sometimes called the pressure flow or the bulk flow method of movement. Now notice, although phloem cells have cytoplasm, they don't have most of the other organelles, they don't have a nucleus. So how do they know what to do? Well, it turns out that the sieve tubes in phloem are associated with something called a companion cell. And if we look at the companion cell has a nucleus, and there's evidence that the companion cell has more mitochondria, even though it may be smaller than the sieve tube. And so the companion cell, for instance, there's evidence that they make ATP, and then they send ATP over to the sieve tube cell through some pores. So companion cells seem to be kind of guiding the sieve tube cells. So let's look at how pressure flow or bulk flow works. And really for this, the key things that we want to keep in mind are how does osmosis work and how does diffusion work? Because this is what we're going to see. So let's look at a diagram of a plant where we have a root, we've got some stems, we've got some leaves. We could have a fruit up here. And when we think about the phloem, one big difference between phloem and xylem is that xylem transports water and solutes that are dissolved in water upwards, mostly from the roots, but like I say, there's one exception. Phloem transports materials up or down. It transports some water. It transports materials dissolved in water, but a lot of the materials that are transported in phloem are organic compounds. And the main organic compounds that plants transfer from one spot to another is one of the sugars that we talked about back in Biology 105, a sugar called sucrose. Remember that sucrose is a disaccharide, and sucrose is made by combining one molecule of glucose with one molecule of fructose. So when we look at phloem, phloem can transport materials in either direction, up or down. But there's one thing that's true when phloem transports materials. Phloem always transports materials from something called a source, which in this example is the leaf, and it always transports it from the source to a sink. So when we think of phloem, movement is always from a source to a sink. Now remember, we had that special term transpiration that referred to the evaporation of water from our leaves. When we look at the movement of materials in phloem, it's called translocation. So we want to keep that in mind. So let's look at what happens here. So we have a leaf that's making carbohydrates. It's carrying out photosynthesis. The carbohydrate molecules are the black dots. So there's a high concentration of these black dots in the leaf where they're being made. What happens is the leaf transports sucrose or some other carbohydrate, usually it's sucrose, but it transports sucrose inside the phloem, inside those sieve tubes. So sugar is transported into the sieve tubes. Now, when we transport sugar into the sieve tubes, that lowers the concentration of water. So what does that do? That pulls water into these sieve tubes. Now, in another area of the plant, like where we have a stem starting to grow or down in the root, we have this sucrose being removed from the phloem. So notice this is a source. This is where sucrose is entering the phloem. This growing tip of the stem or this root is a sink. This is where sucrose is being removed from the phloem. Well, notice when sucrose is removed from the phloem here, and usually the sucrose has to be removed by active transport, but sometimes it can happen through facilitated diffusion or just regular diffusion. When the sucrose is removed from the phloem, again, that decreases the concentration of water in the plant cells around the phloem. So since that decreases the concentration of water, water moves out of the phloem by osmosis. So notice, sugar is being moved into the phloem by active transport at the source. Sucrose is being moved out of the phloem by active transport at the sink. But the water is following along passively in both cases by osmosis. So we have water entering the phloem here. We have it leaving over here at the stem tip and down here at the root. So this means there's a lot of water in this part of the phloem, not so much over by the stem tip or the root. When we have a lot of water in one area and less in another, we can measure the amount of water by something called turgor pressure. So the turgor pressure in this area, and it's always going to be true, wherever we have the source, the turgor pressure is high. Wherever we have a sink, the turgor pressure is low. So when you hear high and low, you should think of our discussions in 105 about diffusion and osmosis. What do we know? Things move from where they're at high levels to low levels. So this means that the material flows in the direction of a sink and it flows from the source. Now remember, these phloem cells contain cytoplasm. So these materials actually have to move through the cytoplasm of the phloem to move. Now, let's look at the one instance where xylem transports materials that really didn't come from the roots. So let's look at xylem and phloem together. And keep in mind, we've seen several pictures of vascular bundles at this time. So you probably remember that xylem and phloem are usually located next to each other. So let's look at some xylem represented by the blue cells and some phloem represented by the pink. So let's look at a leaf cell that's carrying out photosynthesis, making sucrose. So sucrose is pumped into the companion cell, which then gives it to the sieve tube. And as sucrose is pumped into the companion cell and the sieve tube, water enters. And this forces the material in the phloem down, in this case, to a sink. The sink is a root cell. And in this area, the companion cell is removing sucrose from the phloem, and then it's being transported into the sink cell. So up here in the leaf, we're putting sucrose in, water follows passively by osmosis. Water moves down because of that turgor pressure. That turgor pressure means the water always moves from a source to a sink, and whatever is dissolved in the water, like sucrose, will move with it. When sucrose exits, water also follows it. And not all the water follows it in this case. When we look at an area where there's a sink, some of the water that leaves the phloem, this pinkish area, goes over by osmosis and enters the xylem. And then the xylem carries it up. And at some point along its journey up, we talked before about the fact that the water might exit and go into a mesophyll cell and be used for photosynthesis. It might leave the leaf by transpiration. But another possibility is it can move over into the phloem, because remember, the concentration of sucrose is high here, the concentration of water is low. So it can move over to the phloem by osmosis. So again, there's no energetic cost. So some of the water just kind of circulates between the xylem and the phloem. It doesn't really get anywhere. And so this is a good example of how in biology, we often have physiology that shows us a coupling of two different processes that, in a sense, do something different. Here we're transporting water upwards, here we're transporting it down, but we can use this movement of water to make both processes more efficient. Are there any questions about xylem and phloem and how things move in them? No. Okay, I'm going to stop sharing the screen now, and I will let you go ahead and leave for the weekend unless you have any more questions. As I said, I'll be posting plant questions. I'll definitely post them by Monday, and they'll be due not next Monday, but the Monday after, but I'll try to get them up over this weekend. So if you want to get started on them early, you can. But if no one has any questions, you're welcome to go. And I will see you on Monday. Enjoy the weekend.
Have a good weekend.
You too, Corey.
Have a
Have a
good
good
day.
weekend.
Bye-bye.
Thank you. Have a good weekend. See you Monday.
Bye-bye.
Bye.
Have a good weekend.
You too.